Hello, my name is Hazem Kalaji. I'm a professor in biology and I'm working at the Department of Plant Physiology, Institute of Biology, Warsaw Agricultural University in Poland. It's my pleasure today to present for you the equipments which are available for not only the scientists but as well for the agricultural sectors and the commercial sectors who is dealing with the agriculture as well. So we are talking about the farmers who are working in the field or in the greenhouses. These type of instruments allow us to do non-invasive, very fast estimation of the physiological status of the plants. So in a few minutes we can apply any type of these measurements and we can get information about what's going on inside our plants. So as you know, usually we used to bring the plants inside the lab. Now we are free. We are coming with these sets of portable instruments, coming out and travel everywhere we would like to go. We go to the forest, we go to the lake, we go to the greenhouse or to the field. How we can do our measurements really in a very short time, because those measurements are usually done sometimes in 1 second and some others they just do the measurements in a few minutes. So that means in a short time we can get a huge number of information about our plants, and we can then combine these all parameters to get better understanding about what is going on in our plants. So if there is any type of stressors, we call it stressors or distress, that means anything which is unfavorable for plant growth or development, so we can see that because before it's coming to be seen by the naked eyes. So this type of instruments can allow us to predict the stress effects on our plants. Then we can work on to stop what is going on or what is the stressor which can affect our yields or growth of our plants. So the first set which we are going to talk about, it's related to the light measurements, and the first part will be the light intensity measurements by photometers, we call them. Then we have the other part which is related to the light quality, and it's spectrometers. Then we go with instruments addressed to measure the chlorophyll content directly in the field without chemical analysis. And then we go to the set which is addressed to measure the chlorophyll fluorescence. And this is one of the most important, let's say, today around the world among the farmers, the scientists, tool that can give us a very good picture about what's going on with our plants with a very early time detection up to 2 or 3 weeks sometimes. So before we can see what's going on with our plants for a long time, these instruments can discover if there is some changes already started in our plants or not. So the next set of the instruments are infrared gas analyzers. They are portable instruments. We have here 2 instruments, one from PB Systems company from the United States, the other one from Lightmeter. And both are addressed to measure the, we call it plant gas exchange parameters. That mean photosynthesis intensity, transpiration intensity, and stomatal conductance, and the carbon dioxide concentration beyond the stomata. The last instruments, it's showing the effect of all these, let's say, processes which could be measured by the other instruments, and all they are addressed to understand the photosynthetic productivity and efficiency of the plants. So you can see that at the leaf area measurements and the architecture of the plants. So for these purposes, we have a plant canopy analyzer, which give us the ability to make or to analyze the architecture of the plants and then measure the leaf area, which is a result of these higher or lower photosynthetic productivity and efficiency of the plants. So now we will go step by step, one by one of all these instruments to describe how to make measurements and briefly to give some information about how we do measurements, what are the units, how it's functioning, and where or how it could be exploited. So the first instrument which I would like to describe today is the photometers, is the Lightmeter. It's an instrument which allows us to measure the photosynthetic actively radiation. This is the range of between 400 and 700 nanometers. This is the range of the light which the plants can use in photosynthesis. We call it the visual light. And we can measure it by this type of instruments. This type is little old because it has analogy meter. Then we can see here the measurements in the numbers, as you can see here. So to do a measurements, at the beginning we have to check the battery quality, if they are good or not, because if the battery will not be charged, that can affect our measurements. So usually we check the battery, then we can choose that we would like to make these measurements in air, because these producers is giving us the ability as well to make the same measurements for light intensity under the water. But we are not under the water now, we are in the greenhouse and we would like to measure the light intensity in the air. So we put it on the sensor in the air and we choose the range, expected range of our measurements. That will allow us to get better precision of our measurements. So if I'm choosing, for example, 1000, it can show me now about 110. It's not precise. So if I go to 300, and I can read now from the down here scale, we can see that it's more precise and it will be about 117 micro Einstein. Just to remind you that the units which we are measuring the light intensity for the plants, we call them micro Einstein or micromole photon per meter square per second. So to do our measurements, please remember that if you are in a closed place like here in the greenhouse, you have to make first the sensor horizontally. This is why we have here the leveling units. And keep it far from us and from any other objects which can affect our measurements. So we can make now the measurements peacefully, doing in one point, then we go to the other place in the same place here in the greenhouse, and we do many points as well, then we can take the mean value. Because we cannot trust only one measurement, because as you can see, there is various elements above us related to the construction of this building, and then we cannot trust just only one measurement. So we have to do a multi-measurements, then we can take the mean value. The next instrument which I would like to talk about today is again Lightmeter, but it's measured the photosynthetic active radiation energy. And these sensors, as you can see here, is named by that purpose. So we are measuring the energy of the light in the range between 400 and 700 nanometers, again, but in other units. if you remember the first instruments which we measured by the units of micromole photon per meter squared per second, which is micro Einstein, as we said, but in this way, we have it, in these instruments, we have it by watts per meter square. And to make the measurements, first we have the choice on the back side to see what is the expected range of our light intensity, which we have here in this place. So if we have a low light, we can choose from 0 to 19. If we have a mean value light, we can take the other one, which is about 0 to 200 or 199. Then 0 to about 2000 in the third range if we are outside and if it's very sunny day. So usually we run the instruments by pushing this button. We put the sensors again far away from us and we look for the values until it's stabilized, and then we can register this value. Again, it's recommended to make many measurements in the same place and take the mean values, because this is only a point measurements in one place. So it's better if we are giving, let's say, the environmental conditions of our experiments, to make the same measurements in different places than we give the mean value. So the next group of the light sensors is addressed to measure the light intensity and a certain wavelengths. So these type of light sensors, as we showed before, could be addressed only to measure the photosynthetically active radiation, which is in the range of 400 and 700 nanometers. And that will be one photodiode inside, built in this light sensor. But on other side, we can have many photodiodes inside, then we can measure the same light intensity in different ranges. So we can have it, for example, like in this sensor, in the red and the far red region, which is 600 and 660 and 730 nanometers. This ratio is very important for any plant growers in the greenhouses or in a controlled environments, because in the nature, this ratio should be about 1. It's about 1. So if we built our greenhouse with foil or glasses and we added some LEDs or any other sources of light, like the halogen lamps or others. So we would like to check if this ratio stays at the same level, which is about 1. So if we have very high red light intensity, the ratio will be above 1. And if we have very high far red light intensity, the ratio will be below 1. Why it's very important to do such measurements in the greenhouses or in any controlled place where we have artificial lighting for our plants? Because as we said, in the nature, it's about 1. So now, as we found that when you have a very high light, red light, the plants will be stunted, they will be very short, and they will not grow normally. On other way, if you have a very high light intensity at 600, 730, which is far red, the plants will go very fast to grow. That means the elongation growth will be stimulated, and we will lose the fruits or the final yield. So it's very important to keep this ratio at 1 when we are growing our plants under artificial lights. How to do measurements? We run the instruments, and we can see on the back side that there is a range of the expected light measurements which we are going to do. Now I'm expecting here something below 100, so I can take the scale maybe at the first level, which is between 0 and 200. Now on the other side, this button here can allow me to take first the measurements of the red light intensity at 660. Then if I go down, I can take the light intensity at 730. Then I can take the ratio. And this is why I'm interested in. So we go back to the other side. We put the instruments, as we said, far away from us, at the same level of our plants, and we take the measurements. Let me have it now at the red light. Then we have the far red light, and then we have the ratio. And the ratio is about 1. That means these conditions here now in this greenhouse are very similar to the natural conditions outside. That means the growth will be normally. The next instruments which I would like to show you today is related to the light quality analysis. So as we saw in the other light sensors, they can give us the light intensity in a certain wavelengths, mostly used in the plants or sciences related to plants is the photosynthetically active radiation between 400 and 700 nanometers. And as we said, we can get the values either in micromole, photon per meter square, or in micro-Einstein. But the new generation of these light sensors, it allows us to give the light intensity in many units at once, and on the same time, as you can see here on the screen, it can analyze the light quality. So we know about the light intensity in a certain wavelengths. We can see here on this screen the 3 units which are measuring the light intensity, and in the same time we can see the light quality curve. On the other hand, we can get the colors of these light analysis directly on the screen by the use of such small portable instruments, which could be handheld or could be fitted on a triangle or something similar to that. The next instrument which I would like to talk about is related to measuring the temperature of the plants or the leaves. One of these small instruments which is now available on the market actually is addressed to measure the temperature of different objects. It's for industrial purposes, for buildings and others, but among the purposes as well, it could be applied here in the case of plant temperature. The temperature will be related to the physiological status of the plants. So if they are ill or there is any type of stressors there, the temperature will be higher, especially in the case of the lack of the water in the ground. If we are dealing with a drought, stress, and there is a lack of water in the ground, the temperature of the leaf will be higher because the transpiration will be lower. On the other case, on the opposite side, if we have a enough water in the soil and our plants are happy and they are growing normally, so the transpiration rate will be higher. That means the temperature of the leaf will be lower. So let us see what are the values for these chosen leaves here in this case. So it's enough to choose the objects. It could be the whole plants, or it could be the whole canopy, or in the forest, in the field, in the greenhouse. But in our case, we will take only one leaf. I choose the points where I would like to make the measurements, and we take the photo. When we take the photo, we analyze it. By the tools which are coming with these instruments. And then we have the photo in the plants, a usual normal photo. We have the photo where we can see the chosen points where we did these measurements. We can choose the language anyway. And we can see here a report about the temperature coming from these 3 points, which was chosen by me from the leaf. Then we have many other parameters related to analyzing the environments around these objects which we analyzed. So we have information about the ambient temperature, about the humidity, and the distance, and other parameters. The next instrument which I would like to present today is related to chlorophyll content measurements, and the instruments, as you can see here, named as Chlorophyll Continent meter. So this type of meters comes with from different producers with a different scale of the units. The units is the same, it's relative units. So we are talking about if we have high level of chlorophyll or low level of chlorophyll. We don't have specific units like in the chemical methods when we are analyzing our plants in a destructive way, though we have them in microgram per gram, fresh weights or dry weight, whatever. Here we have only a relative units. Why we need these type of instruments? To learn about the chlorophyll content of the plants. And as you know, the chlorophyll is related, its concentration is related to the photosynthesis process, and photosynthesis intensity process or productivity is related to the final yield. So that means if I have a plant with higher chlorophyll content, I can expect that the yield of these plants will be higher than those with lower chlorophyll contents. Usually it's a very simple measurements, and by the way, it could be calibrated with the chemical methods by a curve which we call it a chlorophyll content calibration curve. That means I can take many measurements in non-invasive way using these instruments. Then after taking the instruments on this leaf, I cut it and take it to chemical analysis to see how many milligrams per gram fresh weight, and then make a calibration curve showing what is related relative units to a certain content of the chlorophyll in those classical or SE units. Then when I come back to my field, I just make the measurements in relative units, and then again I put the points on the light calibration curve, and then I know how many milligrams per gram fresh weight inside these plants without doing any chemicals analysis anymore. So usually there is a light coming from one side of these leaf clips here, coming through the leaf, and we have a detector on the other side which is measuring the light intensity which is coming out from the other side of the leaf. The differences between the light intensity from one side and the other side, that is the light which is absorbed by the leaf. And then this light absorbed by the leaf, it depends on how much chlorophyll we have inside. However, we have to be very careful when we are doing measurements on a stressed plants, especially if we have salinity or drought, where the structure, the anatomy of the leaf changes. Then we have a thicker leaf. So despite that, we have less content of this chlorophyll, the value will be higher. And this is what we call it in science artifact. So we have to take care in this way. And in the other, let's say, examples we found in our experiments, that when we have different nutrients deficiency in the plants, these chlorophyll content meters, they don't work ideally. So we have to take care. It's give you a very nice measurements and results when you are doing your measurements on, usually on a plants growing in optimal growth conditions. So to do the measurements, first we have to choose a place where is a spot on the leaf which is representative for the whole leaf. Please remind yourself that the leaf tissue's age is different depending on the place, on the spot on the leaf. So we have here a young tissues. On the other side, here on the bottom of the leaf, we have old tissues, and we have here middle age, let's say middle age tissues. So this is representative for the whole plants. However, if my measurements should be very precise, I would take measurements in the middle, on the bottom, and on the top of the leaves, and I will take the value, the mean value of the whole leaves. Now, if I would like to make measurements for the whole plants, it's better to take leaves which is representing the young leaves, the average or moderately grown leaves, and the old leaves. Then I have an information which is matched or coming from not only one measurements, but different measurements coming from different levels and ages of the leaves. And we should consider as well the side which we are doing these measurements. So I would take, for example, for these plants, at least 3 or 4 measurements from that side on different leaf ages. Then we go to the other side, to the third one, and to the fourth one, because the lightening everywhere in the nature or even in the greenhouse, it's not equal from each side. So let's try to see what the values. We will take here a young leaf, and we make the measurements, and it's 21.8. Then we take average leaf grown, and we make measurement, and it's 44, so it's almost double. And then if we will take old one, as I said, please avoid all the nerves here of the plants, just put it on the leaf. And we take measurements, and it's 65. So 20, 40, 60. In this way, we can confirm that the old leaves, they have more chlorophyll content than the young one. This is why if we would like to make a representative measurements, we have to consider young, middle, and old leaves from different sides. And then if I would like to make measurements or to talk about the chlorophyll content of these plants here in this place, I have to make at least many repetitions for many plants as the same as I did with these plants here. Okay, our next instruments, we call it stress meter or florimeters. It's a type of instruments which allow us to measure the physiological status of our plants, which is related to the photosynthetic efficiency of our plants. And we have two types of these measurements. The first one is after dark adaptation of the samples. So we use this type of clips. And the other types will be described in my next step of this presentation. It doesn't need a dark adaptations of the samples. And both of them, they are right, they are complementary, and they are giving a very important and very useful informations for not only the scientists, but for the farmers, for the growers, for anybody who is dealing with the plants in controlled or semi-controlled or open environments like in the fields. So the first technology, we call it a continuous excitation system. And this is an example from Hansa-Tech company, which is coming as the main florimeters with head sensors that can allow us to give a light intensity here for excitation of the chlorophyll, and then we can measure back the signal, which we call it chlorophyll fluorescence. So if you can see that here we have the firmware and the software, and we establish our parameters, how much and how many light intensity, what is the duration of the excitation, and how many repetitions we would like to have. Usually this type of measurements, take about 1 second, and in 1 second we will have more than 120 points on the curve, which we call it chlorophyll fluorescence induction curve, and we will have more than 70 parameters related to photosynthesis and photosynthetic efficiency of the plants. So if you look here inside, you will see that we have light source. This is a red light, 650 nanometers, and in the middle we have a detector with a filter which is cutting any light coming from our samples, reflected or re-emitted, not reflected, re-emitted from our samples, which will be enclosed here in the leaf clips, above 700. So the plants will see these lights in a minute when we are going to do the measurements, and you can see now here the red lights coming from the head light units. So we will apply this light for excitation of the chlorophyll. So usually you put the head sensor on the leaf clips, which we are using for dark adaptation. And the dark adaptation of the samples here is required because we are measuring, by the use of this type of instruments, the maximal activity of photosystem 2, that means the potential of photosynthetic machinery or apparatus. Then we have to use such type of clips, or the other way that you can cut the leaf and take it into the dark in your lab, and then you can measure there using only one clips, though you don't have to have a big amount of the leaf clips, but it will be, as I said, an invasive, destructive measurements, because you will take the leaf inside the lab in the dark, and then you can measure directly without the lead of these clips. The third way as well, that you can come out to your plants in the night, where they are already adapted to the dark, and then you can measure all or any leaves you wish without the need of dark adaptation clips. So in this is the most recommended way. However, And then we choose, like in the case of chlorophyll continent meters, as we said, a representative leaves on the same plant from different levels. We have here younger, we have young one, we have, let's say here, moderately developed leaves, and we have old leaves on the bottom. So we take measurements from one side, we make them again from the second side and the third and the fourth, because the sunlight is not equal on our plants in the field or in the greenhouse. And again, if we would like to know the photosynthetic activity of our plants here, efficiency of our plants, so we go to make the same on many plants and then take the mean value of these measurements. So we come with the head sensor. After running the instruments, we put it on the leaf clips. We open the leaf clips where already the leaf is dark adapted, at least this spot which you are going to measure, and we make the measurements. Then directly we can see the results on the screen, and we have the main parameters, the minimal fluorescence, maximal one, FO, FM. Then we have the FV, which is the variable fluorescence, and we have the most important parameters, FVFM, which is or should be about 0.83 to 0.85 for any plants in the world. So if we have lower values than 0.80, 83, that means there is some stresses there in the plants. And we can recognize that before we can see any changes by our naked eyes. So this is the advancement of these methods, that we can predict the stress or the changes in the plants, or we can make early detection of nutrient deficiency, water deficiency, droughts, cold, any other type of stressors. Within the 1 second measurements, you can see that we have one measurement each 10 microseconds. That means within about 300 milliseconds, we have more than 120 points that allow us to draw, and the software do that for us automatically when we connect the Flora Meter with the PC. Then we have the curve, which we call it chlorophyll fluorescence induction curve, which is starting from the minimal fluorescence FO and ending by the FM, which is the maximal. And all these changes on these induction curves can indicate or could be calibrated to identify a stressor. Is it salinity, drought, calcium, potassium, phosphorus, whatever nutrients deficiency. The next instrument is addressed to measure the photosynthetic efficiency of the plants, but without dark adaptations. It's enough to have the ambient light, and we can come to our plants to make the measurements in 1 second. So this system we call fluorescence monitoring system, and it doesn't need, as we said, dark adaptations. These instruments giving us information about the actual status of photosynthetic apparatus or photosynthetic machinery or photosynthesis of our plants. While the other one, which I described previously, is giving us information about the potential, the maximal value of this photosynthetic efficiency of our plants. And the differences are something like if we go to the doctors and we feel bad, so he would like to measure our physiological status by the meaning of the heartbeat. So they are doing two types of tests. One when we are in a peaceful sitting way and they are hearing the pulse of our hearts, and then the other one is when we are running on a belt and they connect the sensors and again to analyze our heart beating. So it's a very similar technology. We are hearing the heartbeat of the plants, which we call it the photosystem 2. And again, as I said, after dark adaptation, this is a fixed value for the most of the plants in the world. It's about 0.83, 0.85 for the parameters FVFM, while in this case, for the measurements of PBS2, the parameters which we are getting while we are doing the actual chlorophyll fluorescence values or photosynthesis activity values, then we have values about 0.78, 0.79. And this is the optimal value of the most of the plants. So any values below these values, as we mentioned, that means the physiological status of our plants is affected by any type of stressors. And as I said before, we can see these decrease in this value, which can indicate something which is happening with our plants, but we still cannot see it by our naked eyes. So this is the only methods for early stress detection. Doesn't matter, is it nutrient deficiency or water deficiency as well, or any other type of stressors. So to do measurements, we just go to the plants, we take a representative spot and we push the button. And in 1 second, we have here the values. We have the FS, which is the steady state fluorescence. And in the case of the stress plants, we have higher values. And if they are in a good conditions, these values will be low. The other one is FM BREEAM, which is the maximal fluorescence under the light. And if we have a stress, this value will go lower. And the other values, which is the most important here, is the actual efficiency of photosystem 2, which is PBS2, and we have here equal to 0.76, which is good. So in 1 second, we come back to our next plant or next leaf, and we can save that one, and we can do next measurements. And again, we take older leaves, you push the button, and we save, and we collect our data from one plant. Next plants make many repetitions here in the greenhouse or in the field or in the forest, whatever, and then make the mean value of these parameters, and then we understand what is the physiological status or the conditions of our plants. The next instruments, we call it infrared gas analyzers or portable photosynthesis systems. They are type of instruments which allow us to measure the photosynthesis intensity, transpiration intensity, stomatal conductance, and intracellular carbon dioxide beyond the stomata. The system is based of two units. The main units, we call it the console, and the other one we call it the cuvette or the chamber. It depends on if we are using only a small part while we are doing the measurements, then we call it cuvette. But if we put the whole leaf or the plants in a chamber, we call it a chamber. So this is differences here in the names. In these units, we have the chemical absorbance here on the other side. We have the connections, and we have the main board with the display, which is showing the measured parameters, calculated parameters, and inside we have enclosed the cuvettes where are the measurements provided there. And we have usually in these open systems one channel for carbon dioxide and the other channel for the humidity. So once we connect the instruments to the cuvette here, we run it and we start to see these parameters on the screen. So the symbol of carbon dioxide references is referring for the carbon dioxide in the reference channel. Then in the sample channel where we have here the leaves will be closed now. And then we have the humidity of the reference channel and the humidity of the sample channel. Then we have the differences between both channels. So if I have plants inside and I have light, so I'm expecting that photosynthesis process now will start. That means the carbon dioxide in the sample channel will be lower than in the references one. Then I have the delta carbon dioxide. On the base of these differences here, the value of photosynthesis or assimilation is calculated. On the other side, if my plant is under the light and doing photosynthesis and transpiration, that means it will lose water. That means the humidity in the references channel will be lower than in the sample. And then again we have the differences between the two values here, which is going in the firmware to calculate the transpiration rates of these plants. Then the other parameters are calculated on the base of these two. The first one is the stomatal conductance of the plants, which is related to the transpiration. Then we have the carbon dioxide, the intracellular concentration of carbon dioxide intracellular, or the intracellular carbon dioxide, and which is referring to the amount of the carbon dioxide under the stomata which is not used in photosynthesis. So usually if my plants is healthy and it's good conditions, the value of photosynthesis will be higher, the transpiration usually will be higher, the stomatal conductance will be higher, and the carbon dioxide, intracellular carbon dioxide will be lower. On the opposite side, if my plants are stressed, the photosynthesis intensity will be low, transpiration will be low, stomata will be closed, and the intracellular carbon dioxide will be high. Now we can see here the value for these plants. For photosynthesis, it's about 4.2, which is equal under these slide conditions. Now coming back to this, the second part here of our instruments, as we said, we call it the cuvette. The cuvette is usually, it's like a small growth chamber, and it's equipped with a light meter, as we explained at the beginning of our presentations today, which is measuring the light intensity in the range of 400 to 700 nanometers. Then we have the ability to control the temperature increasing or decreasing by Peltier elements. Then we have the ability as well to control the humidity here electronically, and the carbon dioxide inside the chamber as well, if I'm not using the ambient carbon dioxide from outside. So usually this is one of the most important instruments when we would like to be used, when we would like to learn about the photosynthesis of our plants, which is related directly to the final yield. So whenever you have plants with higher photosynthesis, so you can expect that you will have higher amount of biomass and then higher amount of the yields and bigger fruits, as we can see here. So the correlation, as you know, between photosynthesis and the final yield is very strong, and this type of measurements during the growth difference or various growth periods of the plants can allow us to predict the yield level which we are going to have at the end of our farming and cultivating our plants. The next instruments, we call it plant canopy analyzer. It's addressed to measure the architecture of the plants, and it's based actually on two main parameters. The first parameter is telling us about the leaf area of the plants growing on 1 meter square totally, and the other parameters is addressed to give us information about the leaf angle of the in the plants or the branches. So these two parameters are very important parameters to give us information about the expected yield, because depending on the leaf area and the leaf angles, we can expect our final yield. Is it going to be high or low? So this unit contains actually two parts. The first one is the analyzer itself with the built-in firmware, and then the other side we have here the stick or the sensor, which we have with special lenses here that can allow us to measure the light intensity, and we can have the references value above the canopy, and then we can do the measurements below the canopy, and then on the base of the differences between the reference value and the below the canopy value, the instruments will calculate for us the leaf area index and the mean tip angles of these plants or these branches. So to do instruments, we adapt the stick in this way to have a reference value after running our special protocol here. And we have the bottom. So we take reference value, which is 100%. Then we can see here that we have stars beside these values. So if the star is at the beginning here of this line on the top side, that means I have to do measurements above the canopy. But if the star here appear on the lower line, that means I have to make measurements below the canopy, and now I will do that. And I made a protocol here that I will repeat 3 times my measurements to get better results. So now when I finished, the instrument is computing and it's calculating these parameters. Then we go down, we review the values, and we are looking for the MTI, which is MTA, which is the mean tip angles, it's 69. This is 69 degrees. And then we look for the leaf area index, which is 1.4. That means the plants which are growing here on 1 meter square, if we will cut all these leaves and measure it in a destructive way, the total area will be 1.41 meter square per meter square land or ground. Usually, the leaf area index for the most crops and plants is something between 3 and 6 meter square per meter square. At the end of this presentation, I would like to show you these very nice products from NASA that could be used by any farmer or scientist for very fast detection of any stress which can affect our plant growth. So it's enough to put these glasses and go to the field, to the forest, or in the greenhouse, and we can see the plants that are affected by any type of stresses in a different color than those which are in optimal conditions and good physiological status. And at the end of this presentation, I would like also to invite you to listen to my lecture, which will be a detailed information on the use of this type of portable computerized instrumentations, and it will be addressed for all of them, all of these techniques which I have described during my presentation today, and most of the instruments used in plant biology, physiology, ecology, and agriculture sciences. Thank you.